Hello, my wonderful and lovely humans. So today we are going to be filling out this dihybrid crosses worksheet. So we are going to learn about dihybrid crosses. You're going to find out that it's pretty similar to the Punnett squares that we've been doing, although it is a little bit more complex. So just make sure you take your time. Also, please, if you guys feel like you aren't getting this, realize that this is hard, okay? Even Miss Reyes has a hard time with it. This is not a strength for me. And I definitely did have to check in on how to do these before I did. So again, if you guys find this hard, please don't feel bad. It is hard, okay? All right, so we're gonna go ahead and get started. So let me switch the camera over to my dot cam. And also I'm gonna warn you guys, King Triton is around and you guys know how that goes. So just be prepared for it, okay? So first off, what I want to explain to you guys is that this whole assignment, okay, which I consider the first page, so this page right here, and the second page right here, is going to be worth 25 points, okay? So if you complete all of this, and if you complete all of this, I'm gonna give you 25 points. Now, I'm also not gonna waste this last page here, okay? This last page is for up to an extra 10 extra credit points, okay? If you guys choose to do that, again, totally optional, I will give you an extra credit or 10 extra credit points, okay? Alrighty, but before you guys can do that, you guys have to know how to do it. So let's go through it. Now, just to let you know, we're gonna go this way, we're gonna go this route, okay? And then we're gonna come right back on this side. So I'm gonna go ahead and zoom in because this is teeny tiny. And if you guys have a hard time in class, you're definitely gonna have a hard time now. So let's make sure that focus is in. Oh, look at that. Okay, so what even is a dihybrid cross? Well, a dihybrid cross is very much like a Punnett square, except they look at 16 traits, 16, instead of just one. They are used to work out the probability of the different offspring genotypes. And what's the opposite of genotype? Yeah, guys, it's phenotype. So the different offspring genotypes and phenotypes for each trait. So guys, I made a mistake. Ugh, y'all. It's been a night already. This is the third time I filmed this and I'm not going to restart it. So, because I actually don't have another copy of this. So they're not going to look at 16 traits. They're going to look at 2 traits. Whoops, awkward. Okay, so we're going to look at 2 traits instead of just one. So they're going to work out the probability of the different offspring genotypes and phenotype for each trait. So this diagram is going to contain 16 squares of results. So we must take extra special care and a little bit of extra time when we're filling it out, okay? Alrighty, so now that we've got this done, all right, now that we've kind of talked about it, let's move on to some monster phenotypes. So here we've got 2 traits. Over here we've got a coding for eyes, and over here we have a coding for fur color. So we have a coding for fur color. All right. So we're looking at 2 traits instead of just one. We used to just look at the one. Now we got 1, 2. So we need to look at those simultaneously. All righty. So if we go through this first one, the allele for 3 eyes is going to be dominant over the allele for 1 eye, which means what's the recessive one? Exactly, 1 eye is recessive. So let's go through our, monster here to our first one. And we're gonna look at that first trait here with the T's, okay? So we've got 3 eyes that is dominant. So what kind of phenotype are we gonna see resulting from this genotype? So the genotype we have here is called what? Yes, it's heterozygous. And when we have a heterozygous genotype, it always expresses itself within the phenotype, which is the physical trait, always expresses itself as the dominant trait. And I know I kind of threw a couple of wrenches in those in that understanding for you guys with the incomplete dominance and codominance and all the other ones. However, this problem does not anywhere state that it is a different type of dominance. So we can assume that it's going to be the regular kind. Okay. So don't overcomplicate it by accidentally thinking that this is more complicated than it really is. It's not. It's just that basic dominant takes over the recessive. So because we have a heterozygous trait here, we're going to end up seeing only the dominant trait. And if we look at that dominant trait, that is 3 eyes. So we're going to give this a little monster dude over here 3 eyes. Oh, and he looks so cute, so happy. Okay, so our little monster over here is going to have those dominant 3 eyes. Okay, so the next trait we need to look at, these last 2 letters here, okay, B, is going to code for that fur color, and the allele for blue fur color is going to be dominant over the allele for the orange fur. So we can say that the orange fur is for sure the exactly the recessive allele. Okay, so let's take a look over here. For this trait, this guy is also heterozygous, and as with the first, the dominant trait ended up taking over the recessive trait, which means that this guy is going to have the dominant trait, which is blue fur. So guess what? I'm going to take my little blue marker and I'm going to color this dude in. Now, usually I give you guys extra credit for coloring. However, guess what? Today, not an option. You guys definitely need to be coloring. So if you guys do not have any markers or crayons or colored pencils, pause the video, go grab them right now, okay? We are gonna need to make sure that we color these in because trust me, you're gonna need it, okay? All righty. So now that you've gotten your crayons or colored pencils or markers or whatever you guys need, because you guys have them in the classroom and you guys know exactly where they are, let's look at this second little monster dude here. Again, we're starting with the T's over here, and what we see are 2 of the same alleles, 2 of the same T's, and they're both lowercase, which means we call that what? And there's 2 words to this. Yes, it's homozygous because it's same, and it's recessive because it's the lowercase one. So it's homozygous recessive, okay? So this guy is homozygous recessive for the eyes, and as we can see, the recessive trait that would show up is gonna be that 1 eye. So this dude, I'm just gonna give him 1 big old cyclops eye. Also pretty cute. Okay, so now we're going to look at that second trait, which is that B, and that B is going to code for the fur color. Okay. So once again, we do have 2 smaller B's, which stands for recessive, right? And they are the same. So what do we call it when they're the same and they are the lowercase ones? Yes, it is homozygous recessive. So we have 2 homozygous recessive traits. The first one was for the eye, and now the second one's going to be the fur color. And if we look at the recessive trait for fur color, it is orange fur. So this little dude is going to be orange. All righty. Now, I fully trust that you guys are capable of filling these 2 guys out. So go ahead, pause the video, and make sure you get both the number of eyes and the color of their fur. Okay, so now that we're back from that, and hopefully you guys have these 2 filled out, again, with the color and the number of eyes, let's move on over here. So we've got a bit of a challenge here. We're gonna need to work backwards. So they're giving us the phenotype, and they want us to work out all of the possible genotypes for each monster. So if we look at this guy, we have 1 eye, and it says that this little dude's orange. So let me go ahead and skip ahead here, color this guy in orange, just to make sure I remember. And I'm going to color this guy because he says he's blue. So he's going to be blue. All righty. And again, they look so happy. All right, so this guy's definitely orange and he's got one eye. So first off, we started with the eyes. So let's make sure that we look at the eyes first. So he's got one eye and we know that one eye is the recessive trait. And when we see the recessive trait, we know that the coding or the genotype is for sure going to be the recessive one. And we are using T, so we're gonna have 2 lowercase t's, okay? All right, so this guy has 2 lowercase t's and we call that homozygous recessive, okay? Alrighty, so now that we have looked at the number of eyes, we need to look at the color of this dude. So if we go look over here, the blue fur color is dominant over orange fur, and he has orange fur. We can see that he has orange fur, which means that his genotype must be, exactly, it's pretty much the same as over here. It's homozygous recessive, and instead of using T's, we're going to use B's. So we got 2 little b's or lowercase b's, okay? So this guy's overall genotype for both eye or number of eyes and fur color is going to be homozygous recessive for both traits, okay? Now we need to move on to our little blue guy over here. So this guy is blue, and as we can see, blue is going to be dominant over orange fur. All right, he's got 3 eyes, and 3 eyes is dominant over 1 eye. So we're not as clear cut here with the genotype. So we're going to work it out together. No worries. Alrighty, so first let's start with the eyes because that's what came first. Now this guy has those 3 eyes and that's the dominant trait. So we know for sure this guy has at least 1 dominant trait, and the dominant trait for the eyes was that capital T. So I'm going to go ahead, since I got the space over here, I'm going to write that capital T. Now, there's 2 possible codings for this guy to still show up as blue because he's dominant, right? And as we know, as we saw here, when we have that heterozygous, that guy still looks blue. So that means that we could have homozygous dominant. So we could have capital T, whoops. Or we could have a heterozygous pairing, which is capital T, lowercase t. So either one of these are going to code for a 3-eye color, okay? Alrighty. So now that we got those 2, let's look at the color of this little monster dude. So if we look over here, blue fur is going to be dominant over the orange fur. And once again, because this guy, wait, did I switch the eyes and the blues? Oh my goodness. Okay. So we did eyes, now we're doing color. My bad, y'all. Same thing. But anyways, okay, now we're doing fur color. Now we're switching to the bees. My bad, guys. Sorry, these guys are distracting me. Okay, so the allele for blue fur color is going to be the capital B, and that's going to be dominant over the allele for orange fur. So because this guy is blue, again, there are 2 potential possibilities because blue is going to show up because it's dominant. So as long as that capital or the dominant trait is there, it's going to show up as blue. So we can have either homozygous dominant, or we could have the heterozygous. Okay. So now we have 4 potential ones. However, this guy's genotype is the culmination of 4 genotypes. So let's make sure that we work this out to make sure that we get all of the possible genotypes that they're asking for here. Okay. So I'm going to use some colors here because y'all know Miss Reyes likes her colors. Okay. And I'm going to use, let's see, this really crazy pink color. Okay. So I'm going to start with these 2 capital T's over here. Okay. And I'm using pink. So what I'm going to do is basically distribute that out to each possible genotype for the color coding. Okay. So I'm going to go ahead and boom. So the first pairing that I could get, and I'm going to write that in pink. The first pairing I could get is homozygous dominant for both eye color, or sorry, number of eyes, and for the number, or sorry, for the fur color. So we have capital, 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 capital. Okay. Now, if I go and I distribute that to the second one, okay, so the heterozygous for blue, what we're going to see is still that capital T, but now we're going to see the capital B lowercase b, which is again heterozygous. All right, now let me switch colors. Let's use, I don't know, is red gonna be super obvious? Whatever. Okay, so I'm gonna use red to start the distribution process for the T's over here. And again, the T's are gonna code for the number of I's. So if I go ahead and basically distribute that out, I'll start down here. So we're gonna have a capital T, lowercase t, paired with a capital B, lowercase b. So we have two traits that are both heterozygous, okay? So the last thing that we're gonna do is we are going to have to distribute this out to the last pairing. So that means that we could have a capital T, lowercase t, or heterozygous, and we could have a homozygous dominant, okay? So there are 1, 2, 3, 4 possibilities for this guy's genotype, okay? And those are all over here. And you know what? Let me just go ahead and draw an arrow because again, this is teeny tiny and Miss Reyes didn't have enough space to write that. So that is what belongs right in here, right in this box, okay? Alrighty, so like I said at the beginning of the video, we're going to be doing a little bit of jumping around. So we finished this side now. We're going to go back up here to the corner. Okay, so now we're really going to start looking at these dihybrid crosses, okay? We got a little bit of the taste of having basically two traits in one problem. So now we really need to better understand what Miss Reyes was doing over here with distributing, okay? So we're going to go really in depth here, and we're going to do so by looking at parent gamete alleles, okay? So a gamete is going to refer to the egg or the sperm cell, okay? And the eggs come from females and sperm come from males. All right, so we're going to use a FOIL method to work out the parent gamete alleles from their genotypes. We need to color code the arrows and the words to explain the method. And guess what? Miss Reyes already has. So make sure you guys go ahead and do that on your own. I'm going to walk you through this, okay? So over here, we have 4 circles. And guess what? Hey, that's an egg cell, right? So we have 4 egg cells over here. We need to make sure that we're distributing these out properly. So first, the first letter in FOIL is F for first, right? Appropriate, isn't it? So we are going to start with the first letter of the first trait, and we are going to connect that with the first letter of the second trait. So when we do that, the pairing that we get is going to be capital B, capital Y. Once we're done with the first one, we move on to the second letter, which is O for outer. And what we're going to do is connect the outermost two alleles here, okay? So we're going to have a capital B, and it's going to be connected with a lowercase y, or a recessive I, or y. Alrighty, the next step is going to be the third letter, which is I for inner. Now we're going to move on to the inside, right? So here we're going to do the innermost alleles. So we connect this lowercase b with the uppercase y. So, and B is going to stay first, okay? These are 2 different traits. So B is going to stay first. And yes, I realize that it is recessive, but it's going to stay first, okay? So we have a recessive B and we have a dominant Y. Okay. And that last and final letter, the 4th letter in FOIL, stands for last. Again, really appropriate, isn't it? So what we're going to do here is the last letter of the first trait and the last letter of the second trait. Okay, so if we connect those two, what we get is lowercase b and lowercase y, or 2 recessive alleles. Okay, so now that we've got that worked out, what they want us to do is really practice it and kind of get a hang of it, okay? So I'm gonna still follow this color order that I used up here. So just try to follow along and yeah, do what we can, okay? Alrighty, so the first step is to look at this guy right over here. So this is the first parent's genotype. And if we look at these cells, right? Oh, look, we got some circles over here and we know for sure that's a sperm. So that must be egg cells, right? So this must be mom and this must be dad. So we're gonna do mom together, okay? We're gonna get mom's gametes done. So the first thing we need to do, right? Think of foil is first. We need to start with the first letters of each trait. And that's in green. So what I'm gonna do is connect the first letter to the first letter, sorry, the first letter of the first trait to the first letter of the second trait. And what we get, and I'm gonna write this in green, okay? What we get is that capital D, capital L. All right. Then we need to move on to the second letter, which is O for outer. And that I did in blue. So I'm gonna also do in blue down here. Now I'm going to do the outermost letters, and the outermost letter starts with that capital D and ends with that capital L. So, oh, look at that. It's the same thing as that first gamete. So we have capital D, capital L. Okay. Alrighty, so we're on to the third letter of foil, which is standing for inner. So we're gonna look at the innermost letter or alleles of the traits here. So we're gonna go ahead and connect those two. So we got a lowercase d with an uppercase L. All right. And finally, we move on to the last letter of that foil, and that is in purple. So I'm also going to write it in purple, okay? And last stands for the very last of each trait, right? So this lowercase d is the last of the first, and that capital L is the last of the second trait, okay? So when we put those together, we get a lowercase d and a uppercase L. Oh, look at that. These two guys match as well. Interesting. Okay, so now that we took care of figuring out what gametes mom has over here, why don't you guys go ahead and pause the video and figure out what gametes dad has down here, okay? So you're gonna follow that foil process. Once again, just go ahead and distribute them out. Go slowly, take your time, and make sure you get it right, okay? Alrighty, so now that you've gotten a chance to fill these out, and again, Miss Reyes isn't going to fill it out for you, so you guys go ahead and fill that out. We have basically completed that first page, and that first page basically showed us how this whole dihybrid cross works out, okay? So what we're going to do is move on to the first problem, because when we do this first problem, guess what? It's going to help you to figure out the second problem. And yeah, you guys can get those 10 extra credit points for doing this one. So let's figure out how to do it. Let's figure out how to get those 10 extra credit points. Okay, so we're going to be looking over here. We're going to start on the right here. We need to read through the instructions, and then we basically, it's going to tell us step-by-step instructions on how to really complete this, oh my goodness, complicated mess, right? Again, there's 1, 2, 3, 4. 1, 2, 3, 4. So what's 4 times 4? 16. There's 16 of these. Oh my goodness, we went from 4 to 16. Crazy, right? We're gonna be fine. We just need to make sure that we're taking it slow and that we are checking our work, okay? And again, please remember that this is hard, okay? Do not feel bad if you are having a hard time, all right? Okay. So let's go ahead and zoom in on these instructions, make sure that we understand what's going on here. So let me get a pink marker, which is probably a color that I haven't used quite yet. So let's highlight the important information. So here's our practice problem. We're going to be looking at eyes. So the allele for brown eyes, okay, and we got a capital B, is going to be dominant over the allele for blue eyes. The second trait that we're going to be looking at is the allele for long eyelashes, okay? And that's going to be dominant over the allele for short eyelashes. Okay, so now that we've got that done, let's look at what parent number 1 and parent number 2 have for their genotype. So parent number 1 is heterozygous for eye color and is also for eyelash length. So let's go ahead and work out what parent number 1's genotype is going to be. So I'm going to go ahead and write that to the side because again, we have the space. Let's use it. So if we are heterozygous, does that mean that we match or that we are opposite or different? Exactly. It means that we're opposite or different. So we for sure know what the coding is going to be. And because eye color, we're using B, we are going to basically use a capital B and a lowercase b. Okay. So we got that capital B, we got that lowercase b. And now we need to look at the eyelash length. So it says that she is, or parent number 1 is also heterozygous for eyelash length. And if we look at that, the long eyelashes, the capital L, is going to be dominant over the allele for short eyelashes. So we know that we're going to have a capital L and a lowercase L. I'm going to write my L like that so you guys can for sure see that it's an L or a lowercase L. Okay, so on to parent number 2. Let's look at parent number 2. Oh my goodness, Cheyenne. Now I'm not just fighting Cheyenne. Now I'm not just fighting King Triton. I'm fighting Cheyenne too as well. Okay, sorry guys, it's getting late. Okay, so parent number 2 is also going to be heterozygous for both traits. Okay, so hey, if parent 1 was heterozygous for both traits and parent number 2 is heterozygous for both traits, what does that mean that their genotype must be? Yeah, it's going to be the exact same. So we really don't even need to do the work again because we already did it the first time. So I'm just going to go ahead and copy that down for parent number 2. So this is our genotype for parent 1 and parent 2. So what this wants us to do is to work out the possible offspring genotypes, okay, so the coding, and phenotypes, the physical traits, and then give the expected probability or likelihood that these genotypes are going to occur, genotypes or phenotypes are going to occur for each of those phenotypes, okay? So let's go through a step-by-step process. So the first step here is to make sure, Cheyenne, the first step here is to make sure that we are going to get the parent genotypes, okay? So we need to make sure that we get the parent genotypes, which we just did right over here. And we need to figure out what the alleles are going to be in each of their gametes, okay? And once again, those are those egg or sperm cells. So it's got a little working process here. So we can go ahead and input parent 1 genotype. And we already did the work over here. So all we got to do is bring it on down. So we got capital B, lowercase b, and capital L, lowercase l. Okay. Parent number 2, same thing. It wants us to write parent number 2's genotype. So all we got to do is copy that down, or we could even copy this down because remember, they're basically the same. We've got capital B, lowercase b, and capital L, lowercase l. So once again, these are the 2 parents' genotypes. Now we're going to have to go through that FOIL process to figure out what the gametes genotypes are going to be. All right, so we need to make sure that we're using that FOIL process. So again, I'm going to use my pretty colors because again, Miss Reyes likes pretty colors. And I think, what do we use first? We used green, then blue, then orange. Sorry, green, blue, orange, and then we use purple. Okay, so I'm going to make sure that I keep the same order so that way you guys can definitely track what I'm doing here between this and the first page, okay? So the first thing, right? Why don't we actually write that FOIL acronym down so we can make sure that we're following it correctly, okay? So that is FOIL, FOIL, okay? So F That first F stands for first. So we need to start with the first two letters of each trait. So we go ahead and connect them. That means we're going to end up with a capital B and a capital L. So I'm going to go ahead and put that in that first gamete box right there. All right. The second step of FOIL is that O, and that stands for outer. So that means we need to connect our two outermost alleles. And what we have there is a capital B and a lowercase L. The next one, all right, now we need to move on down to the second allele of the first trait. So we're going to do that one in orange. And we're looking at the third letter over here, which is I, and I stands for inner. So we need to make sure that we connect the inner two most alleles, which are lowercase b and capital L. The last step of FOIL is that L over there, and that stands for last. So now we need to connect the last of each trait. And that means we're going to connect this lowercase b to this lowercase L. So now we've got a lowercase b and a lowercase L over here. So just like the genotypes over here, remember the parents are the same. Guess what? Their gametes are going to be the same. So we can basically go ahead and copy all of this information down here to the parents. So I'm going to pause the video so I can do a little bit of movie magic and get those filled in really quick. Okay, so now that we are back from our movie magic, or I don't know, maybe Shondaland, we were in Shondaland. Okay, so we have finished that first step. We figured out the genotypes and we figured out the gametes for each of those parents. So now we can move on to step number 2. Step number 2 is going to be to write the parent gametes. Right, the parent gametes along the top and on the side of the dihybrid cross. So I'm going to go ahead and zoom out a little here so you guys can see just the whole page really quick. So what this is saying here is that we need to write the parent gametes. So these are the parent gametes. We need to make sure that we write them, fill them in over here. So we're going to copy this information for parent number 1 over here because parent number 1 has those egg cells and clearly these are egg cells over here. So we just need to copy these over here and we need to take these over here, these sperm cells, and copy the gamete genotype information right on down here. Okay, so again, I'm going to do a little bit of movie magic and I'm going to fill these in. So abracadabra. All righty, now we are back. I know you guys don't think I'm funny. It's okay. It's okay. I do. And so does King Triton, apparently. Anyways, so now that we've got those gametes copied on over here, we can go ahead and start to fill out this dihybrid cross. But first, let's make sure that we know what step number 3 is. So again, I'm going to zoom back down so you guys can definitely see what's going on here because it's teeny tiny. Step number 3 is going to tell us to work out the offspring genotypes. Okay, we're going to work out the offspring genotypes by reading across and down, just like a normal Punnett square, except a little bit bigger. So that is step number 3. So we can go ahead and I'm going to zoom out a little bit again. All righty. So let's start with this first eye here. Okay. And since both of my traits here are going to be green, I'm going to make sure that I use a green color just so that you guys can, make sure you follow along here. So again, we start with parent number 1. We bring down parent number 1's information and we're going to start with the B's. Okay. So parent number 1 has a dominant B. So I'm going to write that down here. I'm going to look at parent number 2 and see that parent number 2 has another capital B. So I'm going to go ahead and give that to baby number 1. When I look at parent number 1's genotype for, or allele for L, I see that they have a capital 1. Okay. And I'm going to put that first. And then I'm going to look at parent number 2 and see that they also have a capital L. So I can go ahead and add that in. Now, when I'm looking at this, what I can see is that I have 2 traits and each of them are the same. And what do we call that when they are, first of all, capital and they match? Yeah, that's right. So they are going to be homozygous dominant. Okay. So this is a homozygous dominant trait. All right, we're going to move on to baby number 2 over here, and we're going to go through the same process, except now Miss Reyes has 2 colors that she needs to manage here. Okay, so for baby number 2, we're going to go ahead and take parent 1's information, that capital B, and bring it down over here. Then we're going to look at parent number 2, who's got the green one, and he's got a capital B also. So I'll just go ahead and add that on over here. Now I'm going to look back at parent number 1's second trait, and that is that lowercase L. And hey guys, don't remember, does lowercase go before or after the capital one? You guys are so good. So since this is a lowercase L and I do not know yet what parent number 2 is going to give to baby, I'm going to go ahead and put that L towards the end. Leave a little bit of space there, just in case parent number 1 has a dominant trait. Oh, and look at that. Guess what? Parent number 1 did have, or sorry, parent number 2 did have a dominant trait. So good thing I left that space open because that definitely goes before the lowercase L. So what we see here, again, the first trait, we do see the same letters. So both capital, and therefore they are homozygous dominant. However, now we see that the second trait is going to be coding for heterozygous because these 2 no longer match. Now, don't get the colors confused. Again, I'm just showing you what mom brings and what dad is going to bring. Okay. Alrighty. I'm going to do the rest of these 2 with you guys as well. So again, I'm still managing 2 colors at the same time. So as you guys can see up here, parent number 1 is going to go ahead and donate that lowercase b. And again, I don't know what parent number 2 is going to give baby. So I'm going to make sure I leave a little bit of space in the front, just in case it's a capital B. Oh, look at that. It did happen to be a capital B. Oh, thank goodness. Okay. So I'm going to go ahead and write that capital B. And once I got my B alleles, I can look at the L allele. So I'll go back to parent number 1 and see that I have a capital L. So I can go ahead and write that one first. Then I'm going to look at parent number 2 and see that he also has a capital L. So I can just go ahead and carry that on over here. Once again, please do not confuse these colors for traits. Instead, what they mean is they're showing you which parent donated what. Okay. Alrighty. So that means we can move on to baby number 4 over here. Okay. So I am going to start off with that purple color because parent number 1 has that lowercase b in purple. And once again, because that is lowercase, it is recessive, and we put recessive last or after dominant. I'm going to leave a little bit of space at the front just in case parent number 2 has a dominant allele. And guess what a surprise. It's not like we've done it 3 times already, but parent number 2 does have a dominant allele or a dominant B. Okay. Alrighty. So then we move on over back to parent number 1 for that L trait. And we see that parent number 1 has a recessive trait. So again, I'm going to leave a little bit of space and put that towards the end, just in case parent number 2 has a dominant trait. And again, what a surprise. Do I sound sarcastic yet? Okay. What a surprise. Parent number 2 has that capital L. So I'll go ahead and write that in. Alrighty, so those are the first 4 babies. Now I'm going to go ahead and do baby number 5 down here because guess what? Hey, look, now dad's color is going to change a little. Okay. So instead of mom's color changing like it did over here, now it's going to change down here. So I need, try it and get out of my way. Okay. I need blue and I need green. So again, we look at parent number 1. Parent number 1 is going to donate a capital B. So I can write that in the front. Parent number 2 is going to also donate another capital B. Again, write that. Then I'm going to go back to parent number one. Parent number one is going to donate a capital L. And again, Dominic can go first, so we can write that first. But once we look at parent number two, we see that parent number two has a recessive L. Okay. All right, so that was baby 5, 6, 7, 8, 9. Let's work on baby number 9. Okay, so baby number 9. So we've got mom as green, or mom's first gamete is green, and we are going to have dad's gamete as orange. Okay, so from mom or parent number one, we're going to get that capital B. From parent number two, we're going to get that lowercase b. So now we've got heterozygous going on here. Woo. Okay, back to parent number one. Now we're looking at that L trait. Okay, so we got a capital L up here. And if we look at dad, we also have a capital L on this side. Okay, and now we can look, that was baby number 9, 10, 11, 12, 13. So let's look at baby 13. Did I do that right? Yeah, 13, baby number 13. Sorry guys, I'm not good at that. Okay, baby number 13 is gonna have a combination of green and purple allele. So let me make sure I got those correct colors out. You guys don't have to color code these. I'm just doing it so you guys, again, can make sure that you're tracking, what parent's donating what. But we're gonna start out with that green because parent number 1 is gonna donate that big B. And once we look at parent number 2, we see that it's that recessive B that they are gonna pass on over to baby number 13. Okay, once we got the B's, we can look at the L's. So parent number 1 is gonna donate a capital L, and parent number 2 is gonna donate a lowercase L. All right. So now that we've done at least 8 of these, no, wait, did we do 8 of these? 6, 7. We did 7 of these. Like I said, bad at math. Okay, we got 7 of these done. I think you guys can do these last 9. All right, so go ahead, pause the video, get through these last 9. Okay, so now that you've gotten all the genotypes for all 16 eyes, what we can do now, we've completed step 3. Let's look at step number 4. So again, I'm going to zoom on down so you can see for sure what is going on down here. Okay, so step number 4 is going to tell us to work out the phenotype. We just found out the genotype. Now we can figure out the phenotype. And genotype was that coding, phenotype is going to be those physical traits. So in order for us to know exactly what physical traits we're coding for, we got to know the code first, right? They got to give us the instructions. So first, for each genotype, show this by coloring in the irises and adding eyelashes. All right. So again, we're going to be looking at the phenotype for both of these traits that we're looking at. And guys, I realize we kind of got a little sidetracked, so maybe you guys have forgotten, but the traits that we're looking at are eye color. So you could either have brown eyes or blue eyes. And we're also looking at eyelashes. So you could either have long eyelashes or you could have short eyelashes. Okay. So we're just going to look at like 3 of these. Okay. We're going to look at baby 1, 2, and 1, 2, and 5. Okay. So let's look at these guys really quick and make sure that we can get their phenotype. Okay, so again, they could either have blue or brown eyes. So make sure you guys got a blue or brown colored pen. And they could have either short or long eyelashes. So I'm going to just do that in my regular black pen. Okay. All right. So this first baby has 2 capital B's, which means we have a homozygous dominant pairing. And when we have homozygous dominant, we definitely see the dominant allele. And that dominant allele is brown eyes. So guess what? Baby number 1 is going to have brown eyes. Very good. Maybe I should color that in a little better. Okay. Sorry if this looks black to you guys. It's it's brown. I don't know. Okay, so let's look at baby number 2 over here. So baby number 2, again, has that homozygous dominant pairing. So that means that this baby is gonna have brown eyes also. So now that I've actually kind of looked at the genotypes, it would be kind of boring if I did another brown eye. So let's do a blue eye. Oh no, wait, JK. I can't do a blue eye. Why not? That's right, because I don't have any recessive alleles for blue eyes or any homozygous recessive for blue eyes, because we're not going to see those blue eyes unless I have a homozygous. So maybe you guys will find some over here, the mystery eyes, okay? But anyways, back to this guy over here. So like I said, let's do number 5. Again, we have homozygous dominant Bs. So we know that those dominant ones are going to code for brown eyes. What a surprise. Hey, that kind of rhymed. No, I will not rap for you. Don't ask. Okay. I wouldn't rap for anybody. I couldn't. Nobody wants to see that. All right. Anyways, so the second trait that we're looking at, okay, is those eyelashes, the length of the eyelashes. So long or short, and long is dominant. So as long as we have one dominant trait, we're going to see long eyelashes. So what do we have here for baby number 1? We have a dominant trait. So we're going to see some long eyelashes. And guys, I kind of started this earlier, and not going to lie, these kind of look like spider legs. So Miss Reyes isn't good at them. You don't have to be either. Okay. But we're going to have long, pretty eyelashes. Okay. Well, not so pretty. I don't know. Let's hope yours are prettier than mine. Anyways, so now we can move on to the second baby. So second baby is going to have that heterozygous trait, right? So second baby has the heterozygous trait, which means that we have a dominant allele, and we know that the dominant allele always takes over. So this baby right here also going to have long, pretty, or scary eyelashes. Ah, so good. Or not, in my case. Alrighty. Oh God, that looks so creepy. Okay, on to baby number 5 down here, okay? So baby number 5, if we look at that coding, oh look, it matches that one exactly. So I don't even have to look at it. I know that they have long eyelashes. So I'm gonna go ahead, draw my little spider legs. Alrighty. So you guys are going to go through this. Honestly, when you get to short eyelashes, just make sure that they really look short in comparison to the long ones, okay? But you guys go through the rest of these. Make sure that you are filling in both the eye color and the eyelash length according to whatever you found out their genotype was. So pause the video and get to work. Okay, so now that you've had enough time to get through all of these and make sure that you got the color and that you got your eyelashes, now that you have the time to do that, okay, we've completed step 4. We can move on to step number 5 here. And this one, I am unfortunately not going to be able to do with you. However, I have lots of faith in you guys. You guys are so smart. You guys got this, okay? So step number 5 is just figuring out the probability of each offspring phenotype, okay? So they gave us brown eyes and long eyelashes. So you guys are gonna have to go through this and count how many babies have both brown eyes and long eyelashes. So I know at least I've got 3 right here, okay? So however many of those you got, you're going to fill that in here, okay? And it's going to be out of 16 because, again, there are 16 total possible babies here, okay? So all you have to do is count how many brown eyes and long eyelashes for this first one. Second one, it's brown eyes and short eyelashes. So count how many brown eyes with short eyelashes you have. And just basically follow along, okay? It's that simple. Just count how many blue eyes with long eyelashes and then count how many blue eyes that have short eyelashes, okay? Alrighty, guys. So that is the end. You guys did it. So remember, once you filled this out completely, once you filled out the front, you are technically done. Okay. You have done the part that you needed to do. But if you decide you want some extra 10 credit points, you know, for a rainy day or, you know, for a not so great day, always handy dandy to have in your back pocket, some extra credit points. Make sure you do that last page. Okay. And again, color it in, make it look pretty. You know, it's way more fun when things look pretty. Okay. So I will see you guys on Monday. If I am not there with you, I'm so sorry. You guys know that I had a doctor's appointment, but otherwise, get to work. Bye, guys.